Let me see. Is it working? Okay. So, along with our Tuesday lecture, today we are going to check this paper, Cytoplasmic Anillin and Act to promote RhoA myosin to dependent confined migration and invasion. Now, you guys already know that what is the meaning of confined migration, right? And then let's check the Anillin Act 2 again. So, before starting our lecture, so let's see these schematic images. So first, this is how Anillin Act 2 are working when cells are migrating in confined area. So this is, how can I say this color? This blue color is nucleus. And then this little dim red color is cytoplasm. And then star is anilin. And then this gray is RhoA GDP. Pink color is RhoA GTP. Okay. And then Act 2 is not, sorry, this gray color is Act 2. So Act 2, and this is Act 2, this is anilin. So first, you can start when low HTP is needed to activate myosin activity. Okay, one thing is that low HTP is needed to generate some power. And then before low GTP are generated, they are starting from low GDP. GDP to GTP, this transition is coming from Act 2. And then this this low HTP is sequestered to this is anilin. When you enlarge this green star color, this is the enlarged anilin here. RBD is low A binding domain, RBD. ABD is actin binding domain. MBD is myosin binding domain. So this anilin have three specific domain. So definitely low HTP is binding to RBD. So this is called anilin sequester low HTP. Okay. And then this anilin is has actin binding domain. So they should bind actin. Okay. And then MBD, myosin binding domain. So actomyosin activity is also coming from this anilin. So you can imagine this anilin has actin and myosin as a neighbor. So this low HTP is sequestered, and then when they're released, this myosin, which is binding to myosin binding domain, is activating because they are smallly localized. And then because of that, low A GTP is finally working. So low A effectors, they are working. And then this low A effector increase RhoA and myosin 2 activity, and then they start to migrate. When they start to migrate more, so before saying this, so this initial anilin, this initial anilin is some, as you know, some part, not all, maybe 90% is anilin is localized in nucleus, but maybe 10% is localized in cytoplasm already. So when they are initially squeezed in this confined area, this anilin is going to etch back and front. So last time we just call it ACE, right? Anilin, what is it? Yeah, anilin cortical etch. Not exactly. Anyhow, anilin, when the anilin is localized in etch, we just call it ACE. And then this ACE is also binding with actin, right? So we can see this anilin actin complex are made in back and front. And then because of this scenario is happening. So they are when they're activating in RhoA, and then they can migrate more. In that case, some part nucleus is ruptured. And then suddenly this site inside of nucleus anilin is released to outside of cell, outside of nucleus. And then this release anilin from nucleus also deposit front and back again. So this is called nuclear rupture. Cytoplasmic egress of anilin act 2. Egress means just release. So this anilin is localized in front and back. In that case, also actin 2, same thing happened. So I just focused on previously green color star anilin, but act 2 is also initially localized in nucleus first, and then when nuclear is ruptured, they relate to cytoplasm and then deposit in back and front, right? So once again, okay, when cell need to migrate, they have to activate RhoA myosin 2A activation, right? For that, they need some upstream signal. Upstream signal is RhoA GTP, okay? So RhoA, and then RhoA is increased and myosin 2 activity is enhanced, okay? For doing that, how cell are doing their biological mechanism? First, they have to transfer the RhoA GDP to GTP. So GDP is phosphorylation double, GTP is triple phosphorylation. So they have to phosphorylate again, like ADP to ATP. And then this low HTP is binding to anilin, which is already localized in back and front with actin. And then this anilin also has myosin binding domain. So this low HTP, when they are binding here and they are on bond, and then they can activate myosin 2 activity. And then they go invasive migration again. During that time, they can make so much pressure to the nucleus. Suddenly, nucleus some part is ruptured, and then all act2 anilin, which is inside of nucleus, are suddenly released to outside of nucleus, deposit in cytoplasm, and then localize in back and front. And then they're making more power to migrate. Okay. So now we're starting from figure 5. So first, so previously we only focus on anilin, right? So they say about the act 2. So as I told you before, last Tuesday lecture, why they are focusing anilin and act 2? They are already very well known to divide the nucleus when cell division. Okay? So anilin is also working. So anilin act 2 is kind of a couple always. So that is why they are focusing act 2 here. But they want to say endogenous act 2 and then externally added act 2 with HA, some domain. When they check the act 2 localization, they find not exactly, but here some part, little bit, here some part are localized in cytoplasm. This inside, definitely 90% is inside of nucleus. This blue color is cytoplasmic act 2. So always you have to remember that when people say that, okay, some protein is localized in nucleus, but they are always made in outside nucleus. And then they should go back to nucleus. Right? So this kind of transporting is always happening. So, but most of the nucleus inside the protein, maybe 90% localized. But some part is still, they are already made it and they is made at the moment, or some part, this certain protein is sometimes used in cytoplasm in another action. So they show us that Act2 is also localized in cytoplasm, not only in nucleus, but also cytoplasm. And then similarly, they check lifetime. Lifetime is like RhoA activity. Lifetime is low, RhoA activity enhanced. So what is the wild type and the DH mutant? When they DH mutant, this is not happening. When cytoplasm act 2, is when they are act with what is the RhoA GDP, they need TH domain to bind and then phosphorylate the GDP to GTP. So the meaning of this Act2 DH mutant is they cannot make phosphorylation in GDP. So there is no GTP, RhoA GTP. So that's why lifetime is enhanced, which means RhoA activity decrease. Okay. And then it's a little complicated, but let's see. Anillin Act2 wild type, normal. When they remove NNS, what is the meaning of NNS? NNS is nuclear binding ligand, right? When they remove NNS, they are more localized in cytoplasm, right? As I told you, Act2 anilin, they are actually, when you think about the protein cannot make their decision. They are going inside of nucleus, as nucleus, they cannot make the decision. How they're localized in certain part by this using this NNS domain. This NNS ligand, when they're attached to certain protein, and then nucleus importer, they capture and then they transport this protein inside the nucleus. They should not, okay, I'm going to go there. Not like this. They need some transporter. So as a ticket, they need NNS. So when they remove NNS, Act2 and aniline should be localized in cytoplasm more, right? And then there's why when they single and double all the time, you can see they're gradually increasing, or PMLC. PMLC is myosin 2 activity, right? So they confirm that presence of actual aniline in cytoplasm can make more myosin activity. Okay. And then similarly, they analyze. So blabbing cells percentage is meaning when they migrate in certain small part, let's say 30 micrometer square, definitely when they migrate more, they can have more rupture. Rupture means blabbing cells. So increased blabbing cell is they are more migrating. So even in 100 and 30 micrometer square, wild type 30%. When they remove NNS, they are really increasing. Both NNS increasing because this removing NNS can make those two proteins location inside of cytoplasm. Definitely, they are localized in back and front. They are making more power, right? And then when you think about the aniline only minus NS, you already know that, yeah. But actually, when you remove NNS, the blabbing cell is statistically enhanced, but there's not much statistical change here, but trend or enhanced, right? But when they remove this triple three is that this domain, RhoA binding domain, actin binding domain, myosin binding domain. When they remove this, they cannot make this RhoA GDP, GTP, and then RhoA activation. They cannot do, right? So, so that is why they are losing the myosin 2 activity. So there's less migration. So blabbing cell number is going down. Okay, and then similarly happen. So wild type, you know that the DH mutant, they cannot make GDP to GTP. So there is no low activity, less. And aniline delta 3 deletion, low. When they are doing together, low. This trend. So because of this, blabbing cell number is decreasing, definitely cell entry is decreasing, and then entry time, how much time will it take to go inside of that small part? That time is enlarged, right? They have to search and then they need some power, but they are resisted. And then nuclear rupture, same thing happen. Aniline act 2, when they add more, this EGFP is just like they transfect the cell, just plasmid, control plasmid. And then when they transfect aniline act 2 more, and then probably more aniline act 2, they are localized in cytoplasm, right? because they have endogenous aniline active. They can already migrate in confined area. When they transfect this plasmid more, and then more aniline active is generated. So that is why they are increasing the group rupture because they can generate more myosin activity. And then in that case, in the wild type, when they add mutant form, this three domain deletion and then DH mutation, they are going down because this ACT2 DH mutation, they cannot change GDP to GTP. These three deletion domain is this aniline, they don't have any working. Yeah. So from this analysis, they can make this kind of schematic images. So here, any question? Now you guys clear? Okay, let's say one more time. Oh, okay. Oh, that's a very good question. So actually, this paper, they didn't deal with that part. So you are asking, okay, when they penetrate and then finish this confined migration, and then this rupture can go back to normal, right? Probably this this cell, they have rupture membrane repair mechanism or DNA repair mechanism, something like that. So because of that, this rupture membrane should be can be normal. And then at the moment, from the cell point of view, nucleus is not pushing, right? When they finish to confined migration, now they feel some free. So and then from the cell point of view, they don't need to make some very much power. So that is why, and at the moment, this aniline act 2, they can go back to nucleus. Mm. Actually, they didn't say this kind of thing, but when we see the data, and then from other papers, we can easily imagine like that. But maybe when you imagine further, when some stem cell com- experience have experience of this confined migration one time, twice, three time, fourth time, and then probably this go and back mechanism could be damaged, right? In that case, you can say this cell can be like super migrating cell. And then maybe let's say you are cancer cell, and then when they're confined again, and then probably this anilin act 2 permanently localized in cytoplasm more, and then they can migrate anywhere. Yeah, we can imagine like that. Probably these guys, they are looking at those kind of point. In the case, maybe here they are making one small channel, but one channel they penetrate, and that is free, penetrate again, free, and then when we let cell penetrate this small confined channel like 4 or 5 times versus one time, and then their repair mechanism and the migration speed could be different. Okay, and then last figure here, there previously one time here they show they are adding their cancer cell, fibrosarcoma cancer cell in nude mouse skin. And then they analyze how this cancer cell can be confined in skin area naturally, right? So here, this is a non-confined structure. When they're confined by blood vessel or muscle or collagen structure, and then when they're squeezed inside the blood vessel like this, and then they're confined down to 6 micrometer. They show us. Here, they want to focus on cancer cell invasion. Invasion means when cancer cell migrate to normal tissue, we just call it invasion. But invasion is also same phenomenon of migration. So here, H210, their cancer cell, and then GFP anilin, green color, and ACT2, they are adding ACT2 more, and M cherry, and then vasculature. So let's see. So this is like cancer cell spheroid, and then when they after making cancer cell spheroid outside of outside of tissue, like in what is that, clean bench, and then they inject this cancer spheroid to egg. This is a very well-known ex vivo culture model. So when egg, and then when you open the egg, and there's a certain membrane which is called CAM, and then they inject their cancer spheroid to that KAM membrane underneath, and then you can detect this kind of invasion lively. Okay, so when they track this GFP anilin positive cancer cell, here you can see this is a cancer cell core spheroid. This cell is invasion cell to normal egg tissue, right? So which of course this is the core, this is the invasion area, and you can find this invasion cancer cell, they can have more anilin in cytoplasm. This is nucleus, this is cytoplasm. Cytoplasm is here, the cytoplasm, and then even cytoplasm, they can see that, and then they can find this A structure, right? Here in the edge, they are more green color than the normal cytoplasm. Also here and here, but not all, but some cell. Okay, and then they check again. So this amateur is like just say cancer cell itself. Anilin is anilin. So amateur cell only, cancer cell only spheroid. And then when they added anilin act 2 more, so people can ask why you those authors have to use this control group. Why don't they use this amateur only group? Because as I told you, if you add certain protein by transfection, and then the total amount of the protein should increase, which means that when you compare M cherry only and then anilin act to their mutation, definitely anilin active mutation, they are they are adding more protein to the cell, and the basal level is different. So that's why this M cherry only is okay. M cherry positive protein is only transferred inside of nucleus and inside of cell, and the anilin act 2, they are adding more anilin act 2. And then compared to this, when you guys added mutated protein, what happened? And then in the same, probably similar total amount, you can compare it biologically. So when you check these two things, wild type like this, and then you can see anilin structure here, right? Anilin here. And the one thing is that this cancer cell migrating better to outside, and then which means that they can perform invasion better. But when you see this three domain mutant and then to each domain, so there is no transition from RhoA GDP to GDP by this domain mutation. And anilin three domain mutation, they cannot generate RhoA GDP, right? And then they have small, which means that they cannot invade properly. So from these images, live images, they quantification from the tumor, they have A structure, actin, what is that? Anilin localized in edges, cell edges. They are 50% here. Invasive zone, maybe 60 or 70%. Then cells per metastatic lesion. Metastatic lesion is more invasive, right? More invasive. Their invasive score is going down, this mutant. And then when they check the cell track velocity, the speed of cell, they are gradually going down. When you look at this, when you look at wild type with cytoplasm high of this one, low, depending on this, because when you transfect the cell, you cannot imagine all cells are 100% transfected. maybe some cell 100, some cell 50, some cell 20%, depending on how much of plasmid they are transfected. Their level of this actual anilin could be different. So that's why when they check high and low in cytoplasm, depending on that, they can see some difference. And then mutant form, definitely they are going difference, going down when they're mutant. The cell extravasated. Extravasated is that from the vessel, how this cancer cell is going out. This is called extravasated. From the wild type, 80%. Some cancer cell, when they are penetrate inside the blood vessel, and then such a part they can go outside. So when they track cancer lively, 80% going out here, but only 10% is going out from mutant. So from the images, so this J is like normal type, K is mutant type. So this gray color is vasculature. Vasculature is just blood vessel, let's say, blood vessel in egg. And you check this cancer cell, these two cells from the blood vessel, they're ready to go out, right? But here, they are similarly localized in same level, blood vessel, which means that they are just localized in blood vessel. So this phenomenon, when they track lively, this cancer cell initially localized in blood vessel in same panel, but they are slowly going out, which is called extravasation. So from the M here, also actually this is static images, so it's not easy to say, but when they track this cancer cell individually, some cell go inside the nucleus as go inside of blood vessel, which is in. When they're going when they start to go outside, which is called X exit, when they're already outside the outside the blood vessel, which is called out. So they can see some cell go inside, some cell go outside. They are going traveling, something like this. Yes. So this is the end of this paper. So let's look at some extended figure together. Yeah, we show that. Actually, this is their how they make the plasmid. It's a little bit complicated, but wild type here. Okay. When you look at wild type, okay, just wild type has DH domain. This domain, they are needed to bind with RhoA GDP and phosphorate GDP to GTP. So in the NLS here, black color. So when they cut down NLS, they remove this NLS part. In the DH mutant, they cut down this DH domain here. So they are making plasmid like this. And then after make this plasmid, and this plasmid is transfected to the cell. This plasmid can go inside the nucleus. And then from nucleus, they have this DNA structure. So they are making certain active protein from the nucleus. And then here, okay. Along with that, annealing here, rupture means that, okay, actually like this, rupture, how you analyze rupture in a non-lovely pair? From the NS M cherry. NS M cherry should be localized in nucleus always when they are leaked outside nucleus, which is called rupture. So this is intact. But here, a little bit, they are ruptured like this, right? And then compared to this, they try to go back to the increase of intensity, which is called repair. But still some part here. But there also this high level go down again, and then more localized to outside of nucleus, and then go back again and go outside. Depending on this NSM cherry nucleus intensity level and cytoplasm level, they can mention this rupture, repair, rupture, repair. So when you look at and you track this one, when they rupture, and then this you can see Act2 is little here, right? here, and then they're rupture and then release and then deposit it here front type front part. Okay, and then this is anilin. Anilin has this normal type anilin, and there's here domain like ligand peptide sequence. So when they remove NLS, this is gone. Okay, and when they remove NLS myosin actin binding domain, which is called delta 3, myosin actin binding, and then NLS, this cut this part and they're making very short. Okay, this is how they are making domain. That is combination. Okay, let's see. So from the main figure, they cannot have proper space to add these images. So here they add it. So wild type, like this, when they remove NRS, they're more deposit in cytoplasm. Act 2 also, AH Act 2 is more deposit here, cytoplasm. They both, yeah, anilin, Act 2, deposit cytoplasm. Wild type here, delta 3, no anilin, less anilin here. And then PMS level also detected as less. So their quantification. And then these their images. So you can imagine they are checking this one in live. So when you check the website, you can download and see the movie. This normal cancer cell, they are migrating from the core to the edge. Wild type, core to the edge. But when they are mutated, this migration definitely decreasing, right? So this is like not this is static images, but from the beginning, maybe beginning they just show like this here, the edge, like that. But this wild type, they are migrating this part like that. And then you can see this also migration, invasion. Here, very less. So they are proving their concept. And finally, actually this same schematic images, but more like concise schematic is applied in main figure. That is another one. Let's see. Okay, first, nucleus limited is at entry and migration in confined. Initially, nucleus is pressured in confined area. And then initial anilin and Act2, some part they are already inside of cytoplasm, 10%, 5%. So they are localized back and front. So back and front accumulation and contractile happening. And then when they migrate a little bit, nuclear envelope rupture, right? And then and then from the nucleus, anilin Act 2 is released, and they are deposited cytoplasm. And because of that, this anilin Act 2 is localized again in rear and front area, and then actual RhoA anilin cell motility is happening. So from the images, RhoA GDP, phosphorylated RhoA GDP by cytoplasm Act 2, and then this RhoA GTP is cytoplasmic anilin recruitment. RhoA GTP is binding to anilin, but this anilin is already localized in front and back. Okay, and then when the anilin has all the things, actin, RhoA GTP, and myosin, so we can say that this anilin is kind of sponge. They can binding everything, anilin, myosin, and anilin as a scaffold. From the scaffold protein, actin is binded, myosin is binded, and then RhoA GTP is binded. So there is kind of core center. And because of that, RhoA GTP is activated, and then they can make cell migrate better. So this one, initially like that, and then they're compressed, and then this edges, and then when the rupture, they're making more power. So when you look at this initial part, the A structure, this A structure, right? Green star anilin is localized in cell membrane here. And then Act 2, low GTP to low GTP. And then this low GTP is binding to anilin. Anilin already have myosin here, yellow color, and actin here. Yeah. And then this anilin, myosin, actin, low HTP, they are localized in cell membrane here. So they can, this binded low HTP is released, and then they are activating myosin 2. So when they highlight this area here like this, anilin as sponge, actin, myosin, RhoA binding domain, they're here. And when this RhoA GTP is released here like this, and the RhoA, when the RhoA is binding with RhoA effector, then they are generating myosin 2 activity. Okay, so this is the end of this paper. So the message of this paper is that they look into very much about this confined migration. The confined migration concept is like 15 years ago, they are published in Science. But this is their just finding. Okay, oh, cell can confine even smaller size compared to nuclear height. They can go. It's a very fascinating phenomenon. And then people want to know that how cancer cell can migrate this kind of small area, which kind of mechanism can be involved. And after finding this mechanism, the next thing is that, okay, if we block this A structure, this anilin cell membrane binding, cell edge binding, when we block it using certain like drug or some other physical therapy, and then we can decrease cancer cell invasion, right? This is one thing we can get. The second thing is that from the stem cell point of view, actually from the stem cell also can migrate like this, right? But from the stem cell, they are confined in small blood vessel and they try to find damaged area and then they can go. And then when we increase the ACE phenomenon, and the stem cell can find much faster the damaged area. Therefore, so that is why from the cancer, from stem cell point of view, when we increase ACE phenomenon, we can expect better tissue generation. So always cancer cell and stem cell, their their trend is opposite. Okay. So any further question? So I mean, perfectly understood. Okay.